Hi, my name is Kanishka Manna. I am a second year bioinformatics PhD student at the University of Arkansas Little Rock and University of Arkansas for Medical Sciences. So today I will be giving a short presentation about my poster entitled Proteogenomic Analysis to Identify Acquired Resistance Specific Alterations in Melanoma PDXs on MAPK inhibitor therapy. So in order to treat melanoma, current therapeutic approaches mostly focuses on small molecule drugs that target activating protein mutations in pro-growth signaling pathways like the MAPK or mitogen activated protein kinase pathway. The benefit of having the MAPK inhibitor therapy against melanoma has been limited to approximately 50% of patients with the activating BRAF mutation. Here we have to keep in mind that each type of therapy has a particular clinical application and benefit and is ultimately associated with primary or acquired resistance. For MAPK inhibitors, it is acquired resistance. To better characterize the mechanisms of MAPK inhibitor in melanoma, we have utilized patient-derived xenografts and applied proteogenomic approaches using genomic, transcriptomic, and proteomic technologies to detect unique RSAs or resistance-specific alterations and detect therapeutic vulnerabilities. Now, I would like to highlight a very specific challenge associated with proteogenomic applications that comes at the level of data curation, which enables accurate multi-omics data integration. So the primary objective of our project is to present a proteogenomic approach that uses custom curated databases to identify unique RSAs in melanoma PDX models of acquired MAPK inhibitor resistance. And in order to do so, we have used the NRAS Q61L melanoma PDX model, with resistance tumor were developed following treatment with MEK inhibitor. So this is the proteogenomics pipeline for the identification of unique RSAs and detection of therapeutic vulnerabilities in melanoma PDXs of NRAS Q61L model. Before we continue with this pipeline, a bit background about the PDX, what is patient-derived xenografts or PDX. So it is basically tumor fragments derived from NRAS Q61L metastatic melanoma tumor, which was transplanted subcutaneously in mice, and each mice received a single tumor fragment. In figure B, you can see the study design over here. There were 4 resistant models, R1, R2, R4, and R5. These were collected on days 161, and a vehicle not MEK inhibitor treated was collected on day 126. The PDX tumors were later chopped, and the DNA, RNA, and protein extraction was done by following the standard protocols, followed by downstream analysis like the genomics, transcriptomics, and proteomics analysis. In data curation part, somatic mutations were identified by using MUTEC2 and STECLA2 variant calling algorithms. Furthermore, these two algorithms, the results from these two algorithms were intersected to identify the variants within each tumor. And custom reference genome was created using the BSAFE tool, while the annotation was done using SNPFF software package. So the proteins were identified by searching against normal reference databases like UniProt-KB or the newly created custom reference database. So moving to the results part, now in this figure, which shows the PDX tumor showing similar patterns of somatic mutations with the West data, where you can see various levels of characterization based on the variant classification, variant type, SNV class, and top 10 mutated genes. On the left-hand figure, this is an upset intersection plot, which shows all genes, which compares basically all the genes of detected mutations among the MEK inhibitor and vehicle-treated models. About 250 mutated genes were shared by all four resistant, but not the vehicle, okay, tumor. Now, here we have the Pearson correlation of transcriptome and proteome data identifying the common RSAs. From figure A to D, you can see the log-fold change values for each resistant tumor compared to the vehicle is correlated for RNA-seq and proteomics data. So you have over here on the x-axis protein fold change, and on the y-axis you have the gene fold change. And each data point over here is defined as a detected protein with associated gene. Okay, the dotted line represents a fold change of twofold upregulated and downregulated positive and negative values. Okay, the figure E and F shows the upregulated and downregulated molecules at both the transcript and protein levels across resistant tumors relative to vehicle control. Here we have 23 molecules which were consistently upregulated and downregulated. So once again, figure E and F, E is upregulated, F is downregulated. So in this figure, the RNA-seq and proteomics data were searched against the custom-generated proteogenomics databases assembled via the West data. So figure A over here shows the log to normalized values for differentially regulated proteins and associated gene transcripts. Now here only the amino acid change for each protein is shown. You can follow the Z-score to see it. While the figure B and C shows the expanded view of the RNA-seq data along with the West variant calling files. The RNA-seq coverage tracks for FBXO38 protein and the NRAS protein, mutated both. So finally, in conclusion, we have applied proteogenomic approach to identify unique RSAs or resistance specific alterations in MAPKi inhibitor resistance PDX tumor models. We have achieved this with the help of NRAS Q61L melanoma PDX model, which showed acquired resistance to MEK inhibitor treatment. Also, I have stressed out the need to create custom proteogenomic databases for this kind of project involving PDX tumors. Finally, our data also highlights the utility of using both the standard database and the custom proteogenomics database to identify the RSAs and the detection of therapeutic vulnerabilities across the PDX tumor. Future developments of this project includes the extension of the multi-omics platform across a variety of genetically diverse melanoma PDX models, which may be resistant to MEK inhibitor, other MAPKi inhibitor therapies, and which will support the identification of newer RSAs for future therapeutic development, particularly in detecting the target signaling pathways. Also, another major development includes the development of interactive HTML outputs for multi-omics data analysis, which will support proteogenomic studies. So that's the end of this poster presentation. I would like to thank my viewers for listening to my talk. Please feel free to reach me via the emails mentioned in the slide and in my poster. You can also look the paper by scanning the QR code or clicking on the DOI link. Thanks.